So instead of a data-driven rich topic, what we're going to have here today is the opportunity to discuss the concept. And the concept, I believe, is an important one, where we want to be able to take the tools and the developments on the genomic side of DOE and start to apply them to questions and concerns on the environmental side, particularly on the climate side of DOE. And it's not trivial, and so after I got the replacement notice for the canceled speaker, I got a couple of gray hairs in the whisker to try to get a presentation together quickly. But I sent out an email, and this email went out to a consortium of people that I work with on the climate side, and I was amazed at the response and the excitement that there may be a new opportunity to galvanize these two seemingly disparate sort of groups into a common cause, into a grand challenge. And so all of these co-authors that you see here have actually contributed directly to this presentation, okay? So I feel more like an ambassador than a presenter. And so we've seen this concept before of trying to go from genomes to climate models, and this is off of the DOE BER website. And so the idea is sort of there, and the concepts are there, and even the language, and I think we've all seen the text before, right? So we want to be able to take modern molecular biology techniques, we want to put them into the use to study ecosystems, and I think more importantly, we want to be able to predict the response of these ecosystems. But what I think is unique, okay, is that text that you're now looking at was actually derived 22 years ago in a report from DOE before BER was even BER. So this concept has been around for a while, and we're just now starting to address it. And it makes sense, okay? If we look at a heat map of the northern part of the hemisphere with the warm colors representing increased carbon stocks, it's very clear that we have a tremendous opportunity to look and to understand how carbon cycling is going to affect our processes of interest, okay? And here's a point that I think is extremely important, is that if we look at these warm colors in the northern part with the boreal and the Arctic ecosystems, there's twice as much carbon in these stored carbon stocks as there presently is in the current atmosphere. And so the models don't have this, right? So the question of concern is if we have warming and climate change, are we now going to open this system up so that we have increased microbial respiration, and now we have a major problem, or at least we have a major sensitivity in the climate models as they currently operate. So this is the key question that I think we can begin to put these sort of grand challenges in and apply these technologies that we have in the genomic sciences. So I'm heartened to see the most recent call, okay? And the most recent call from Joe Graber's group is that they want to know about microbes and microbial communities and carbon cycling. But for some reason, we don't have these same calls in plant biology. And so the change in community dynamics is important in understanding carbon cycling. And so we need to understand how we can start to rectify this missing element. And so when I asked the climate modelers, what's the problem? How come we're not doing this? And there's a question of scale. So this is the first problem that we deal with at this sort of modeling is scale. And so you can imagine that if you're talking about plants at this scale, and this is a half-meter plot from the Arctic, and if you understand the genomics, you have a systems model where you can actually go from genes and you can go from metabolized proteins to traits and the integration of those traits to phenotype. How is that going to help you as we start to get the landscape scale? And then the climate modelers are working at this level and above. So this is the challenge. So when a climate modeler is working from the top down, we need to know how processes at this level are going to inform the next and the next. And you can think of this as a hierarchical structure. So this is, in a nutshell, sort of the grand challenge. And to give you an idea of the complexity of the scale, this is a project we have in the tropical systems, and Eldridge gave a great intro to this. This is actually his research institute in Stry. And here's the idea of scale. So just imagine if you know the genomics or have a systems model of one of these tree species. There's 157 tree species per hectare, right, in this area. So how do we take that information and how do we make this relevant to the ecosystem? And how do we use this to predict community composition change in a meaningful way? Now, one thing that's missing from that clip is there was actually a pan of our faces. So the scale is so shocking that the person who was working on this from me, Mark Schuster, actually put this clip together. And yes, we were amazed. And so this is even a climate modeler, Jeff Warren. This is myself. The systems modeler is the most dumbfounded of us all. And then, of course, even Anthony Walker. So it's a shocking thing to go up this 240-foot gondola and experience this. So what do we need to do, and what are some of the other challenges? And one is community integration. And so what you're looking at left is, as a system modeler, we just sat down with Paul Hansen, we brought out a schema for a model design. And the idea here is that we want to be able to communicate with the climate modeler so that we know what trait we want to perturb will actually make a difference. And so this is just a start. Of course, the other issue is these larger system models have 1.5 million lines of code. I mean, it's in Fortran, which almost nobody in systems biology actually codes in anymore. So there are certainly challenges, but it brings to question is, what do they want? What do the modelers want? Again, they want to go across scale. They want to be able to take information from genomics. How does that relate to trait variation? How does that trait variation inform higher levels of organization? And this is essentially what they want. And what they currently have, right, is very coarse-defined plant functional types. And so these coarse-defined plant functional types, is it a tree? Is it a shrub? Is it a grass? Is it C4? Is it C3? Very coarse definitions of what the plant is like. And then they go through these competitions, and then that's iterated through decadal timescales. So an error, right, at one level, 100 years later becomes a major sensitivity in the model. And so the idea is that if you understand traits, and you can actually define these traits through genetics and the distribution of these traits in sort of a continuous scale, that you would then increase the predictive capability of plant functional types and these changes. So we're going from plant functional types to traits, and how those traits mediate who wins and loses under changing climatic conditions. And so that's what I view as a major opportunity. So what do we have on the genomic sides that we can leverage, okay? And we've talked a lot about the best association population. And this is the popular work that's been going on in joint with BASC and JGI. And Steve DeFazio just recently did an association genetics approach on bud flush, and there's been a lot of discussion on bud flush in the previous session yesterday. And the idea is that if you increase growth duration, right, you're going to increase height. So of course these guys are looking at this in terms of BASC, how do we increase plant productivity? But that same trait, right, could be useful from an ecological perspective. You can imagine if the plant's growing longer, it's acquiring more energy, it's bringing more productivity into the ecosystem. So these same traits that we're looking at from a bioenergy perspective may at least give us not just the technology, but the actual trait that could be interesting from a climate perspective. So we've gone into, I think, a couple of times of the BESC association, but through the help of JGI, we have we're getting to 1,100 resequencing genotypes. I think they're at 900 now. They're planted in 4 common gardens. 3 are pictured here. And they're replicated in each of these gardens. And what Steve is showing quite clearly is that there are some loci that are directly correlated, okay, to bud flush. And not only that, but if you look at the QQ plots, you have a few alleles which are above the Q score, above what's expected by random. So adaptive genetics, right, and association genetics has now given us advances, which I think we can apply. And there's also other algorithm development. I'm just going to talk about a few. One of these, of course, is coming from cancer biology. And so we've seen a lot of talks where we use the WGCNA algorithms from Steve Horvath's lab. He's at UCLA, and we started collaborating with him a few years ago. And the idea is that you take these genes and then you do these co-expression networks. And then from these co-expression networks, you do other types of supportive omics and biochemistry, and then you start finding candidate genes of interest. And I think the people in this crowd are very familiar with this very simplified system. But one thing that we often miss when we're applying these algorithms is that you can start to scale up. And you can start to scale up to trait and even phenotype. And actually, Steve did this in the original algorithm because he's interested in cancer phenotypes. And so there's a lot of work on biclustering algorithms, but some of the most simple are simply taking the eigenvalue of each of these modules and then correlating to the changes in trait of interest, okay, or the condition of interest. So some of these algorithms can be applied. There's another good example from the Broad Institute. This is Justin Lamb's stuff. And what Justin did is he took human cells, and he used biomolecules to perturb those cells, and he had the control state. And he made these expression profiles. And he put these expression profiles into a database, and then he would take his biological state of interest from the humans, get the cancer profile here, or whatever profile of the biological states of interest, and scan this compendium. And through this, he's actually connecting drugs, genes, and disease. We took a similar approach and used his algorithms, which, by the way, he pretty interested in, just thought it was funny that we were applying these algorithms to plant biology. And what you can do is you can take these modules, these network modules, and start to correlate the phenotype. And again, this is correlation. This is not mechanism. But we can go from these sort of modules and we can start defining through eigenvalue correlations what state the organism was when we took the transcription profile. And we can find that these hub genes actually enhance the predictions that Justin Lamb is making and using this compendium database. So it's just an example of how we can use modern algorithms that we develop again on this genomic side and start to apply them to, say, predicting plant phenotype and phenotypes that are of interest to community composition change. Now, in order to apply this, of course, and I think everybody here is very familiar with this, is we need quality reference plant genomes. And JGI does this better than anybody else, hands down. And it's through this work that we're starting to move forward, but we also need ways in which we can model advanced genomic data. And so I was here Monday presenting a K-based workshop. And in that workshop, what you can see is we can quickly go from association genetic results, and you're going to see this in a minute, where you can highlight the genes that are highly correlated to your trait of interest in an interactive graphic. This is all real-time video, by the way. And then from this interactive graphic, you go into the database, into the K-based database, and through that you start interacting with diverse data types, and then you start getting immediately information on how these genes are connected in pre-computed co-expression networks, ontology enrichments, gene enrichments, and so forth. So we're now beginning, right, on this side of DOE to produce the computational infrastructure by which we can apply advanced modeling techniques. So together with JGI, this is a very powerful resource and something that we can begin to apply to other areas of DOE like climate. So now I want to transition quickly into opportunities that are currently out there on the climate side, which we can then begin to extend this dialogue into areas of interaction. Okay, one of these is Stan Muschlager's work at NG Arctic, and this is a large project. And one of the key questions they have on the plant side is that in the Yangtze River, for example, in 1966, this is sort of the distribution of woody plant material. Right, and as we get into, what is it? I'm sorry, 43 years later, you can see that all the tundra places are now occupied by shrubification. They call it shrub expansion. So the consequences of this at the ecosystem scale are immense, right? Because you're changing the amount of energy. So instead, they call it albedo, the climate modelers. So instead of white reflecting coming off, you have this shrubification, which is now increasing the amount of energy coming into the system, and you have this positive feed-forward loop where now you may have more warming, making that recalcitrant carbon now susceptible to microbial degradation. So this is one question of interest. And of course, Stan wants to know why these plants are adapted to increasing abundance in these perturbed sites. This is a frost cycle, and then you can see through time how this shrubification occurs. So what are the genes and what are the mechanisms controlling expansion into this area? How can we predict which plants are going to occupy this space? Here's another example by Lin-Hung Gu, and again, this is from SPRUCE. Lin-Hung Gu's a climate modeler, and he felt that there was a sensitivity, or I should say a bias, in prediction of CO2 through the tropical regions, and this is at the global scale. What Lin-Hung found is that as CO2, this is a cutout of a leaf, as CO2 comes into the system here, as it comes into the leaf stomatal pore, it actually has to traverse here, and there's a bunch of resistances that are here that are usually calculated. If the models don't calculate that correctly, you have a complete bias, and that's what he's finding with his results. As he compares at a global scale the amount of global primary productivity, you have this bias instead of a flat line. The difference between models that have this mesophyll conductance and models that don't are different. Okay, so this is another area. So what are the traits behind this? What are the traits that govern mesophyll conductance? Is that change leaf architecture that actually actively transport CO2 if there are such mechanisms? Here's another example from Ben Turner. Now, Ben Turner was featured from SPRUCE. He's a scientist there. And what Ben found is he attributed phosphorus availability to species composition. And this is really kind of neat. There's a fault line here, and the difference in phosphorus availability is what he believes is driving the species composition. Now, you can't bring this up without talking about plant-microbe interaction, and I'm part of a plant-microbe interaction SFA, but it's also my extension to say, hey, I'm in the microbial genomics section here, and I'm trying to put an olive branch out there. But the point of this is that phosphorus availability is driving plant plant distributions, we need to start thinking about these organisms as interacting genomes. And I think this is clear, and DOE recognizes this. Even on the genomic side, there's a large plant microbe interaction SFA. JGI is doing a lot of sequencing for this. And we need to think about this as affecting the plant as the extended plant phenotype. And these are famous words from Jeff Dangle. Okay? So just as another possibility to move forward. Now this is the last part of the talk, and I want to introduce you to a system that's in the SPRUCE system. And this is in northern Minnesota. It's a joint project between DOE and the United States Forest Service. And it's really unique, okay? This is bog system, and these bog systems occupy about 10% of the Earth's surface, but they have about a third of the Earth's stored carbon store. So a very critical ecosystem if we want to understand carbon cycling at a global scale. And what's absolutely fascinating from a plant biology perspective is this keystone species, this sphagnum, which is really critical in determining changes in community composition. The way it does this is it has cation exchange capacity where you have polyuronic acids which take on cations for nutrients, and then hydrogen comes out. And as the hydrogen comes out, it acidifies this to about 3.5. So the idea is that this highly acidified environment now helps this plant to outcompete vascular plants from coming in. Okay, and it's this process where you see these mummies, okay, these bog mummies. And actually this guy over here, I think, was reported to the homicide department. Actually, it was so well preserved. Okay, and so it's this process which is storing, okay, carbon. In our case, we haven't found a spruce mummy, but we did have a troublesome log, which is quite old, and I thought that was interesting to put out there. But the magnitude of this project is really something else. Here's a prototype at Oak Ridge before we went to Minnesota, and here's the scale of this. For those of you who know program manager Dan Stover on the climate side, he's a relatively large guy, and this is just, we can use Dan as sort of a reference for the scale here, and it's quite impressive. As we move out to Minnesota, this is where we are now. And so in Minnesota, we have these mammoth boardwalks, which I've never seen in an ecological experiment in my life. And from these boardwalks, we can get to a number of rings, and they're highly instrumented in the fact that you can look at CO2 and methane and try to evaluate the productivity of the system. The idea is that there's going to be warming and CO2 perturbations here. So not only will you be able to do observational studies, but you're going to have an experimental system here too. It's supposed to be a long-term project, 10 years or so, and so there's Paul Hansen, the PI of this project, encourages collaboration. Here's sort of an aerial photograph. Here's the bog itself. This is called the S1 bog, and here are the chambers which are coming in place now. It's really quite impressive. I talked to you a little bit about the importance of this system. I've talked to you about one pathway that could actually be very relevant in going from genes to pathway to ecosystem level response. And there's also another one, and this is a plant microbe interaction. This is really unique work where they're looking at endophytes, okay, so bacteria that actually go into the plant, in this case sphagnum and the hyaline cells. And in these endophytes are methanotrophs, and the methanotrophs are oxidizing the methane, and they're providing a direct source of CO2 to the sphagnum. And they estimate that sphagnum, 15 to 18% of all the CO2 fixed by sphagnum is coming through this association. So again, the questions could be, what is warming? What is CO2? How is that going to affect this interaction? How is that interaction then going to cascade and affect higher level processes of biological organization? So I want to end by a quick use case, okay? And this is, again, just an extension of a concept. And there's probably 10, 20 better concepts out there, but the idea is to get discussion moving. So how does this operate now? We have a simple system. In this case, it's the spruce. I put this out as a cartoon where we have our sphagnum, we have our shrubs, we have our trees. And what the modelers want to know is they want to know how this is going to change through time under various climate scenarios. And they do this by defining very coarse plant functional types, which we talked about before. In this case, it would be tree, shrub, C4, C3, nitrogen efficiency, so forth. And then through this, they make growth predictions. And then this is going through decadal time steps, okay? And so air here is propagated. And it goes into one of four models. In this case, it's the vegetation model, and that gets fed into the community land model. Now, the community land model is what DOE is proposing as the major model for what we work in. So the question is, how do we improve this sort of scheme? And I think we have a number of opportunities, one of which is the genome sequencing and association studies. So we can take this onto a population scale. Again, this gets back to what the modelers want. They want continuous variation. They want better They want better descriptions of how genes are affecting traits and the variation at these different species level and population level. So I think resources here that have been developed can directly apply to that. Two is we need to characterize and update how those traits are moved from where we work at this side of DOE to the climate model side of DOE. And one way to do that, I think, would be to extend a trait variation service. And this is possible in a K-based cyber infrastructure system. So you can imagine at this point in time that if you're interested, say in sphagnum, that you could have a genome sequence, you could have association genetics, you could have the resulting phenotype, and you could feed this all the way in through a service that would actually give you genes contributing to traits and the variation of those traits into a version service which a climate modeler could then just go into and fetch through an API the most recent trait characterization for these plants. This could be an ongoing, updated, continuous endeavor. And so that's sort of the idea of where we can start moving things forward. I want to leave with some very simple conclusions. I think the challenge is clear, right? We have all of these carbon stocks. We don't know how they're going to respond to climate change, right? And we have an opportunity. We have a major, major investment in the genomic side of DOE. And we think we can bring these tools and these systems to bear on questions of climate. But the question is how to move forward. This is just a very, very pie-in-the-sky introductory talk on how we can start doing that. What really needs to happen is we need to continue to break these stovepipes and work across communities. We need to meet that challenge. And second is we need to get involved. And so if you're interested in participating in any of these climate projects, even if it's on the microbial side, you should contact either Chris Schatt, who's over here. He does a lot of the microbial dynamics in almost all of these systems. Myself, Stan Wuschlager, Paul Hansen, and get things moving. So with that, I thank you, and I thank you again for letting me substitute in for the speaker.
Thanks. Dave, that was a great talk. Thank you very much. I just want to make the point from Washington, D.C. that, first of all, predictions are hard, especially about the future, but I see this as a really high priority back east. First, as Dave nicely said, there's enormous scientific opportunity here. This is not an area that's been really investigated at any great length before, so there's a lot of boldly going into the unknown here. And the other is I can't not imagine that some fairly heavy-duty decisions are going to end up in the future being made based on some of these models. So if you don't need additional encouragement, keep in mind that participating in what Dave talked about, contributing to this kind of science, may have enormous impacts down the road.